Good morning, everybody. My name is Keir. I'm a new group leader at the BioHub, and we're pursuing a holistic picture of infection from the moment a virus invades a host cell and onwards through all paths that lead to organismal health or disease. And today I'm going to show you essentially all unpublished data just to sketch out where we're trying to go, and I'm looking forward to engaging with this community, which is new to me, over the years to come as we try to make this happen. Just to give you some context for the system, which is a little hint showing you here, as we all know a little too well, we are swimming in a dark space. There we are, a vast sea of viruses. And thanks to Joe and many others, we've gotten pretty good at identifying new virus sequences just about anywhere we care to look, whether that's in a human, a bat, or a whale. We can dig out all kinds of fascinating and terrifying new virus sequences. But where we've struggled a bit more is in trying to make functional guesses about those virus sequences. And that holds even for new variants of viruses we already know a lot about. In order to understand what those sequences mean for their hosts, we have to do experiments. And so one way I think we can gain some traction here, gaining functional understanding of how viruses are impacting their hosts, is to carry out virus discovery in an experimentally tractable host context where we have a built-in set of tools we can use to study any of these new viruses that we're discovering. And so I'm pursuing this goal in this animal here, illuminated in all of its splendor, the zebrafish. And the reason why I think this is a compelling system to pursue virus discovery and experimentation is, one, it goes from a single cell fertilized egg into a free-swimming animal that's small and transparent in just 3 days. And that allows us to scale multiple levels of biological organization from the subcellular to the organismal, all in living animals. And this has, of course, made it a compelling system to address all kinds of biological phenomena. So there's a lot of tools that have been built to manipulate this system already. And despite the fact that we diverged from a common ancestor with zebrafish roughly 500 million years ago, they actually have a pretty similar immune system to ours. And that includes both innate immune cells like macrophages, which are these red dots that are peppering the animal above there. In addition to those cells, they also have adaptive immune cells, B and T cells. And so, but it's not just the cells, it's also the signaling programs that are providing antiviral defense, which are mainly driven by interferons, just like they're helping us out. And so with all this packaged in this tiny transparent animal, this system is poised to tell us some interesting things, I think, about host and virus biology. At the time I started my postdoc, there were no viruses of zebrafish that were known, and so this looked like a good opportunity to try to fill this gap. And so the way that I went about finding viruses of zebrafish is by tapping into the interferon system itself. And so this is just roughly schematized here to remind you that upon detection of a virus or something that looks like a virus, host cells will turn on the expression of interferons. Those proteins will be released and signal through neighboring cells to induce the expression of hundreds of genes called interferon-stimulated genes that collectively provide an antiviral state to those cells. So I thought I could turn this into a visual system, taking advantage of the transparency of the animal by just inserting a fluorescent label into this signaling program. Among these interferon-stimulated genes, you would get glowing GFP. And that would allow me in an organismal context to just screen for animals that were exhibiting some kind of antiviral response, cluing me into the potential presence of viruses. So I built these fish. I took a highly induced interferon-stimulated gene, ISG15, fused to GFP, along with a GFP marker that's constitutively expressed in the heart to allow me to distinguish transgenic animals. And so here they were swimming around in their dish with their glowing green hearts, but no green elsewhere. And I started wondering where I should go to try to find viruses that would infect zebrafish. And it turned out I didn't have to go anywhere at all before something surprising started happening, and that's that animals just in typical laboratory rearing contexts started glowing green seemingly spontaneously. I didn't add anything to these petri dishes. I could just see green animals, and they look something like this under the microscope with glowing green signal in the intestine, which is that tube-like structure down the center of the animal there, but also in other epithelial cells. So what's going on here? This is just a laboratory environment. I tested the hypothesis that there was some undescribed virus in this system causing these animals to turn green by just taking entire animals that look like this, which are large on this screen but are actually quite small. So you can take a whole animal, grind it up, take all the RNAs in there, and through sequencing and de novo assembly, look for sequences that look like viruses. So I did that, and what came out was a brand new picornavirus. And it infects mostly the gut, but also occasionally makes its way to the central nervous system, including the brain, which is similar to its relative on this tree of picornaviruses, including poliovirus. So this hints at this system being adaptable to study parallel processes of host and virus biology by throwing all the tricks of zebrafish experimentation to interrogate these interactions. And I'll come back to this virus in a little more detail on what we're doing with it next. But first, this was actually just the beginning of the viruses that we're finding in this system. And so by turning into other contexts and conditions, more and more viruses have started to emerge. And I'll just tell you about one other context today, which is looking at adult animals and just looking for the same kind of thing, which animals are glowing green. So to do this is not exactly what zebrafish was designed to do. So most people study zebrafish in this early larval stage, which included those images I showed you earlier of a glowing green fish. And they do that because they're small and transparent. Once they become adults, they're large and beautiful, but opaque. And so, and just to give you some sense of the scale, the difference here, this is roughly scaling those images based on the size of a larvae, where I found the picornavirus compared to these adults. But could we use the same kind of sentinel system to identify other contexts of infection? To do that, I just turned to genetic tricks of zebrafish by making pigmentation mutants that essentially eliminated those nice stripes and gives us some visual access to interior tissues of the animal, including the gut and circulation. And so again, just looking at the same reporter, started looking for variation in the amount of GFP I saw in adult animals swimming around in their standard laboratory environments. And already, once again, there's clear examples of animals that are either not exhibiting any apparent antiviral immune activation, and then some that are clearly very green. And I'll zoom in on a bizarre-looking structure here in the jaw, which really clued me into something potentially weird going on. As you can see, so that lobular structure there is all in the lower jaw of these animals. And so because this was such a distinct signal, I dissected out jaws that looked like this and sequenced that whole tissue and again looked for virus sequences in that tissue. And what came out of this was hundreds of different virus transcripts. None of them were the picornavirus. All of them came from a new pox virus. And so just like Sonali's talk, I'm going to swerve here from RNA viruses into giant DNA viruses. And this new pox virus from zebrafish jaws, which I'm calling jaw pox for now, its closest relative here is the record holder for genome sizes of viruses that infect animals, this carpedema virus, roughly twice the size of CMV at 500,000 base pairs. But of course, it's a pox virus, and so it's connected in this tree to other interesting viruses in the family of Poxviridae, including, of course, the causative agents of smallpox and monkeypox. So we can take now these sequences that I found in just RNA-seq data and try to put this back into the visual picture of where actually is this virus infecting the animal. So here what I've done is I've just taken a slice of jaw tissue and stained it for RNAs coming from the virus. So here, those are those green dots, and all the other gray dots are just nuclei. And what you're looking at, I'll zoom in a little closer here, is a stratified epithelia. So it looks like these viruses are restricted to these epithelial layers, but really only the most superficial layers of these epithelia in the jaw. And so what this means for virus transmission and why these viruses are hanging out in these cells but not others, and when does this change, these are all questions that we hope to tackle now experimentally. So we've got two very different viruses, at least now, including a coronavirus, small RNA virus, and a supertanker pox virus with hundreds of different genes to look at. And what's emerging here is that zebrafish and fish in general are telling us that the same families of viruses have been locked in conflict with the core vertebrate immune system for roughly half an eon of evolutionary time. And so what I think we can start to address by studying these infections in zebrafish is to find out some really core elements of what host virus interactions look like with this tractable system, but also potentially uncover some new evolutionary innovations about how hosts have learned to overcome viruses and vice versa, and to increase our knowledge about general strategies of host virus interactions and outcomes. So I told you that I found these viruses in normal laboratory environments just by sequencing zebrafish tissues or whole animals. And it turns out one of the strengths of zebrafish is people have been sequencing zebrafish animals and tissues for a long time, not looking for viruses, but they didn't do anything in particular to enrich for viruses, and I didn't do anything in particular. But that begs the question that can we go back to this massive resource of all this effort people have put into sequencing zebrafish to learn more about these viruses that I've found so far, potentially discover new viruses altogether. And so essentially what we'd like to do, and what we're starting to do, is to dig into this collection of publicly available RNA-seq data sets, which at the moment is around 50,000 data sets total, and that number is of course growing with time, to screen through those data just like I showed you earlier for both detecting these viruses, the coronavirus and pox virus, and potentially discovering new viruses, and seeing based on which tissues and cell types and experimental contexts we see viruses in different levels of them, what can we learn about in terms of their tropism and what they're doing to the host biology. And ultimately, taking any sequences we're discovering here and reanimating them for new experimental tools, essentially. So can we just keep building our toolbox of viruses to tap into this host virus biology. And so I'll just give you a glimpse of some progress we've started to make here, thanks to a stellar data scientist at the BioHub, Duo, and I saw his name tag out there, so I think you might be able to ask him about this later. And he's already managed to screen through 50,000 data sets for our pox virus and some others, and already there's more than 2,000 data sets that contain this virus. And so it just gives you a snapshot of the range of potential biology we can explore here and help sharpen our hypotheses about what we want to test next. And so just to give you one glimpse of what that looks like, this is a single data set. So on the x-axis, these are all different zebrafish tissues that this study ended up sequencing. On the y-axis is the level of pox virus that was in those tissues that we identified. And so each dot is an individual RNA-seq data set. And from here you can see, again, the jaw seems to be particularly highly infected, as well as other surface structures, including the scales and gills, but much less so in internal tissues like the intestine and the liver. And it also turns out that this virus correlated pretty strongly with the level of antiviral gene expression, telling us that, again, this isn't just some kind of inert thing passing through, but there's a real dialogue here. And so ultimately what we want to do now is start to understand what's driving this dialogue, what's important for it, what are the various potential outcomes, and how can we tackle host virus dynamics with these tools. So I'll jump now back into RNA virus world a little bit to tell you a little about the dynamics we're exploring with the picornavirus. So in this experiment, what I've done is infected animals intentionally, no longer looking for spontaneous animals swimming around in a dish, but taken virus particles and added it only to naive animals and followed them over time. And so this is just a very coarse longitudinal look. And in this case, what you can see is something interesting, is that there's an antiviral response and the site of infection during early infection in the gut there. That's that little dot of green in the intestine. Over time, that response grows, expands, and intensifies. But then later, these animals turn off this response, and also there's no longer virus around. So in this case, we've got some examples of some dynamics in which the animals are infected, but they clear infection over time, and this tracks with the activation of the immune response. So this kind of classical look at of infection and response. And so what we want to do is start filling in this cartoon version of these dynamics to give some quantitative meaning to what these distributions look like over time. So what is the immune response? Which cells are engaged in this response? And how does that change over time, and based on the location of where the infection is taking place, and so on. So one of the tools we can use to get at that is again, turning to one of the strengths of zebrafish, is we can take an entire organism, which is unique among vertebrates, and dissociate it into single cells to apply single cell, for example, sequencing technologies to get some information about which cells are responding to infection or contain infection, and we can do this over time. So I'll tell you just about a snapshot, one glimpse of this approach. And so here we're looking at that map of all cells that came from a single animal. And this is just roughly about 9,000 cells, but it turns out that we can capture around 100 different cell types, so who knows if there are 100 cell types that make up the entire animal at this point, but it's pretty close to other estimates of what that might be. So here, labeling just a few of them, we can pick out intestinal cells and also immune cells. We can break this down based on their responses. So now I'm showing you that same map comparing uninfected animals with ISG15s, their antiviral gene expression. On the left, there's very spotty to no expression compared to virus-infected animals on the right, where we see pretty broad, but not in every cell type, antiviral immune response. If we break this down just on the immune cells alone, we can see some interesting variation, with macrophages showing a strong response, T cells essentially silent here, but also some other intriguing things come out of single-cell sequencing of zebrafish, and that includes immune cell types that we haven't yet seen before. And so this is this question mark, which we're currently exploring exactly what these are, but they look something like an innate lymphoid cell, although it's not yet clear. But now what we want to do is to label all of these cells and look at them in real time. We can take all these snapshots with single-cell sequencing, but what we want to look at is how these things are changing in real time. So we want to take all these different cell types and have them labeled, in addition to labeled viruses, and follow them in living animals. So here's just one glimpse at the kinds of things we're trying to approach with this. So in this is an animal in which all the macrophages are labeled in red. Again, this image we showed earlier. And the antiviral immune response is in cyan. We can zoom in on the site of infection here in gut and watch these dynamics play out over time. So we watch these macrophages crawl around and interact with the infected gut and occasionally gobble up what may be an infected intestinal cell. So this is just 4 hours of time. The entire course of infection, of course, lasts around 5 days or so in this case. So what we want to do is expand on this picture, look not just at macrophages, but all immune cells, and look at the cells that are actively infected over time across the entire organism and across the entire course of infection. And the way that we're going to do that is by teaming up with Loic Royer's group at the BioHub, who's built custom light sheet microscopes and taken and developed some impressive image analysis tools, which I think are too impressive to really show up on this presentation at the moment. Seems to be not quite slowing or, fast enough. But what you're sort of seeing here is every single cell of the, of the organism has been tracked over developmental time. So these are living animals. They can track every single cell in that organism and follow it over time. And so now what we want to do is apply these microscopy methods to following infections. So we're going to subset our tracking on the immune system and the cells that are infected, and we're going to use that to interrogate the progression of infection in living animals. Okay, and we think that by doing that, we'll be able to develop new models of the immune system and its operation as a whole. So not focusing on just one region, but understanding the immune cells, where they come from, and what the others that aren't responding are doing over this course of time, and how that turns into a general rule of immune system action. And by fusing this together with sequencing, single-cell sequencing technology, we think we'll be able to piece together whether or not we can distinguish cell identity just from cell migration alone or cell activity. And so I've told you a little bit about these diverse and growing group of viruses that we've been discovering in zebrafish, and a little bit about the tools that we're using to try to experiment with these viruses, and ultimately our goal of getting to these larger understandings of the systems level operations. So I'd just like to thank Duo, I mentioned, is on the data science team at the BioHub, along with my fledgling team, Fitz and Deepika, and thanks for showing up today to hear about some primordial ideas.
Okay, use the microphones if you can.
Well, really cool talk. What does the virus or the viruses do? What are they doing to the fish? Are they pathogenic, or and are they also spreading in your dish?
Yeah, those are good questions. So the pathogenesis question, these have been flying under the radar, and it turns out not just in the laboratory I was working in, but all over the world. So there's 2,000 data sets there, as I mentioned, for the pox virus. So what I think happened is there's very few, but there are some data sets that come from wild fish, wild zebrafish, and so what it looks like is these are common viruses of wild zebrafish that have come along with them and spread into research institutions around the world. And so I think they're not drastically pathogenic, but some of the sequencing data has started to tease apart certain situations in which they're correlated with disease. For example, in the pox virus, they're they reach very high levels in animals with skeletal defects, which people have associated with certain genes. So I think the picture is going to become more clear in the days to come, but they're not so far drastically pathogenic.
Let's go to the back. Let's go to the back first and then back to the front.
That was really fascinating. Thank you. I guess piggybacking off of Melanie's question on your point, in a lot of mouse work, you find mouse pathogens when you start working with immunodeficient mice. If you co-tank your immunodeficient fish with your wild-type fish, do they get sick? But my main question was about, does everybody in the dish get sick? Does everybody have it? Does all the fish have your picornavirus, have your jawpox, or are there special fish that you can figure out why they maybe don't get infected?
Yeah, so for the first part, I can make immunodeficient fish and test them explicitly to see if there's any difference in virus levels, and in that case, it does work. For example, if you knock out the interferon response, the viruses go up by a lot. So just that very coarse level, yes. The second part, I think the the snapshot tells us that not all animals are infected at once, but I think there's dynamics there where there is clearly transmission and so I think it's going to be interesting to try to understand how this is maintained at the population level and to have finer grained ability to determine when an animal is infected or not without killing it and sequencing it. And so I think we're hoping to tackle those questions. I think they're really interesting.
Thank you.
It was a fascinating talk. Thank you so much. I really enjoyed it. Have you looked at the virome of, like, for example, interaction of these picornaviruses with pox viruses, or like the microbiome evolution in the presence of these viruses in these fishes?
Yeah, I haven't yet. I think that's an interesting, fascinating
It would
question.
be cool.
Yeah,
like how the microbiome shifts like what happens with these viruses in zebrafish.
Yeah, so the microbiome world of zebrafish is a little more mature than the virus world. And so thankfully that gives us a lot of material to work with. And so we're starting to talk to some of the labs that look at the microbiome. And for example, if you look at germ-free animals, does that change the niche of the gut altogether? And so,
or like interaction of picornaviruses with these pox viruses, like.
Yeah, I
know.
think that might come out of the sequencing survey as well to look for correlations between microbes.
Let's go in the back here.
Hi there, Keir, that was amazing. Super cool stuff. I had a question about the ISGs in the fish. Do you see orthologs to common mammalian antiviral proteins? Are you seeing a whole treasure trove of like new functions? Have you looked?
Yeah, that's a great question, and I have started to look. I'm very interested in the evolution of that program, and there is a core set, so for roughly you could say a third are all from mammals are present in fish, but that means, of course, 2/3 or more is potentially new functions, and so I think that's the stuff I'm more excited about, but so far we've been focusing on the mammalian counterparts because we know a lot about those.
One more in the front, and then we'll move on.
Could you say a little bit more about what you were showing at the end about the tracking of the immune cells, or what you're planning there? And particularly, can you track the different immune cells and know which they are and know anything about the state of those, or what's the potential for that?
Yeah, there's a few strategies that we're thinking about taking here. One is we can label specific subsets of cells, and that's, for example, the macrophages I showed in that video. And we can do the same kinds of things for neutrophils, B cells, or T cells independently. But what we're starting with is to use a generic tag for all immune cells and to follow all of those at once and see if we can break down, based on migration or other kinds of interactions, what the cell identity might be or cell state. So we're coming at it from both sides and hoping that hoping that we'll learn something.
All right, let's move on. Thank you, Keir.